Welcome, everyone. Thank you so much for coming to our TYC workshop on the very topical question of modeling phase separation. We had kind of short time. We organized it kind of last minute, and which was really catalyzed by Jeremy Smith coming to do his sabbatical in the UK. So welcome, Jeremy. And then we decided to build a small get-together one afternoon to discuss this question. So we'll have 3 talks. We'll start with Thomas Michaels, and he'll give us a bit of an introduction. Then Jeremy will speak a bit about kind of functional aspects of the phase separation in biology. And then we'll finish up with Gerald, who will tell us a bit about her latest work on reentrant behavior of liquid phase separation. And I mean, it would be really great to keep this as open and collaborative as possible. So I do invite people to just unmute themselves and ask questions at any point if they feel like doing so. Also, you can type questions in the chat and I'll read them in the end, or if I see them during the talk, I will do so as soon as I see them. And then in the end, we'll have a brief panel on some outstanding questions in the field. So let me introduce first Thomas. Hi, Thomas. It's great to see you. So Thomas is actually one of our own now. He's a new lecturer at UCL in IPLS between physics and Faculty of Life Sciences. So Thomas comes from Cambridge, where he did his PhD and fellowship. Then he did a postdoc also at Harvard with Mahadevan. And now we feel very privileged to have him in our own environment. And I'm really looking forward to hearing what he'll tell us today about spatiotemporal control of filamentous protein aggregation. Thomas, please.
Well, thank you very much, Angela, for the introduction. I'm really, pleased to be part of UCL now. Also, such a lively environment, particularly in the context of life sciences and physics of living systems. And I'm also very delighted to be here today. So the topic of my talk is going to be a little bit around asking from a physical point of view, how can we use liquid like organelles or compartments as a way to organize or control biochemical reactions. And I'm going to talk about this idea in the context of a specific example related with controlling filamentous protein aggregation in space and time. So as part of my talk today, I have been told to give a little bit of an introduction into liquid organelles, what they are, and what is the field like in this area. And so, of course, I'm going to take a little bit of a biased view because I'm a theorist and a physicist. So I'm going to focus a lot on the physics and how the physics, determines the behavior of these organelles. But of course, the field is very broad. And if you are interested in this topic, you should also follow talks by experimentalists who also take a kind of very different angle from mine. So the usual starting point of a talk on liquid phase separation is to show a sketch of a cell that you can find in probably many biological textbooks. And in such textbooks, you typically see the cell as being surrounded by a membrane, which is made of proteins and lipids. And the cell is organized in its interior with a lot of organelles, which are there to organize in space several activities. Typically, you would think that many of these organelles are surrounded by a membrane, and indeed, this is the case. For example, the nucleus is surrounded by a membrane, which contains, for example, genetic material, mitochondria, etc. Nevertheless, it turns out that there are a lot of organelles which are not surrounded by a membrane. So these are called membraneless organelles or liquid condensates, and there are really several names for these guys. Here I'm just showing a couple of examples of such organelles in cells, such as the nucleoli, for example, P granules, cagal bodies, P bodies, and the list is increasing over time. You can find these organelles everywhere in cells. They are, for example, emerging prior to cell division. For example, in the example of P granules, which you see in the second picture, the red one, there you see a C. elegans embryo that is just about to divide, and you see these yellow dots, which are these liquid organelles that form and then move in one part of the cell prior to cell division. These organelles have also been shown to emerge, for example, as a result of cellular stress. They can organize protein material, they can localize genetic material, and in general, they function as microreactors that provide distinct environments for performing biochemical reactions. And here I'm showing you a very nice video where you can see what these liquid organelles are doing. So they are called liquid organelles because they share most hallmark properties with actual liquids. So they have a well-defined surface tension, a viscosity. When you bring them together, they fuse, they undergo ripening, Ostwald ripening, for example, and so on. And in this video, you see very well how these droplets are fusing and then relaxing to a spherical shape. They undergo ripening, all things that you would expect liquid objects to do. Now, of course, the key question in this area is no longer, at least, to find new organelles, but to ask what are these organelles useful for? So how is the cell benefiting from having these organelles? And from a physics perspective, and again, from my point of view, this is really probably the most exciting angle to take. This, you can view these condensates as microreactors that host a certain set of biochemical reactions which are associated with some form of function or dysfunction. And the way they do that is by providing a scaffold that, for example, can concentrate the reactants which are called the clients. And so phase separation can affect the outcome of these reactions in a variety of ways. So for example, you could concentrate the clients, hence speeding up the reaction. If you have multiple components, you could co-localize these multiple components in a specific environment, or you can prevent these components from interacting with each other. You can change the chemistry, so the chemical reaction rates, for example. You can play with the rheology, etc. So these are all various parameters that can be changed by the presence of these liquid condensates. And here is a very nice picture from a recent review article that shows the repertoire of activities that these liquid condensates could do or can do in living cells. And for example, one big interest here is in the control of nucleation and polymerization of filamentous structures. So for example, functional filaments like actin or tubulin, or also pathological structures like, for example, amyloid fibers. And here people have shown that the presence of the liquid condensates can localize the aggregating monomers, can concentrate these aggregating monomers, hence acting, if you wish, as a controller of the aggregation reaction. Another very interesting idea is the control of biochemical reactions, for example, enzymatic reactions. And in this context, I should say that I've recently become very interested in understanding this aspect, and I've started a very nice collaboration with Jeremy precisely on this point. And there are in the field really a lot of other very nice ideas. For example, a very beautiful one is this idea of buffering concentration fluctuations due to the fact that if you have varying level of a certain protein that phase separates, the concentration in the phase separated compartment is constant, and so different levels of the protein are going to translate into smaller or larger condensates. So this is a way, for example, of buffering fluctuations. And so as part of this talk, I said, I wanted to give a broad overview of the field, but at the same time, also I wanted to cherry pick one specific topic that I worked on to really show you in practice what you can do from a theoretical perspective in this area. And so today I've decided to talk about the specific aspect of the biopolymerization controlled by the presence of liquid condensates. And so in this context, I'm thinking about functional filaments like actin and tubulin, but also and primarily about pathological structures like amyloid fibrils. So for the people that are not familiar with amyloid fibrils are a very generic type of filamentous structures. They are characterized by a very specific cross-beta sheet architecture, which has these strong hydrogen bonds interactions. And what is remarkable is that there is really a wide range of apparently unrelated peptides and proteins that can form these amyloid structures. And the main reason why one would like to study their aggregation process, it's because of the link with disease. And there are really a lot of diseases, including Alzheimer's, Parkinson's, and type 2 diabetes, which are linked with the formation of these aggregates. And so here the idea is to say, can we use liquid condensates as a way to control the outcome of this pathological aggregation reaction, and in particular to control the toxic effects? So for example, reduce the number of aggregates that we form, or localize their formation in a certain region. So rather than having these aggregates forming everywhere in the system, can we generate, for example, a condensate that would make this aggregation happen mostly inside the condensates? And so in this manner, we could, for example, prevent these aggregates from interacting with sensitive parts. And so you would effectively reduce the toxic effects of the aggregation process. And so the key question here is whether weak protein interactions, such as the ones that derive phase separations of these condensates, are sufficient to make these aggregates form mostly inside the condensates, and what are the physical parameters that control the partitioning of the aggregates inside the liquid condensates. And so to answer this question, I have combined the theory of protein aggregation kinetics with the theory of liquid-liquid phase separation. And so I'm going now to introduce two topics, or kind of as two steps. One is the theory of protein aggregation, and the other one is a liquid-liquid phase separation. And so again, I've been told to kind of focus a little bit also on the basics, so I will try to give you a little bit of an introduction into both areas. So in terms of kinetic theory of protein aggregation, the way people usually start studying this is by listing the variety of microscopic steps by which these aggregates can form and grow. Okay, so we are thinking about linear filamentous structures. And so these structures typically form always by a primary nucleation step. So whereby monomers spontaneously come together to form the smallest stable fibril structure. And then this fibril structure is able to grow by recruiting further monomers in a process called elongation. And this elongation happens at the ends of the aggregates. Now, primary nucleation is just one way in which you can form new aggregates, because, for example, aggregates could fragment. And so by fragmenting one aggregate, you get two aggregates. And so you have increased the number of aggregates also in an exponential manner, because from two aggregates, you can then form four, and so on and so forth. So this is another way in which you can form new aggregates. And if you want to be very general in this description, then you also have to account for processes that generate aggregates from a combination of monomers and aggregates. And one such example is surface catalyzed secondary nucleation, whereby the presence of existing filaments can catalyze on the surface the formation or the nucleation of new aggregates, a little bit like in heterogeneous nucleation. And so with these steps, we are really covering the set of all possible mechanisms by which you can form aggregates, either from the monomers alone in primary nucleation, from the aggregates alone in fragmentation, or from a combination of monomers and aggregates in secondary nucleation. And so now, having this list of steps allows us to write down a dynamic equation for how the concentration of aggregates of a certain size changes as a function of time. And the concentration here, I'm denoted it with FTJ, so where T stands for time and J is the size of the aggregate, so how many monomers are part of this aggregate. And you can ask, how does this concentration changes as a result of the different steps? And so this looks complicated, but in fact, it's just a bookkeeping exercise. So for example, if I'm looking at the first line of this equation, all I'm saying is that if you have an aggregate of size J and you add a monomer M to it with a rate constant K plus, and you take into account the fact that you have two ends, then you end up with an aggregate of size J. But of course, you have to do this bookkeeping exercise correctly and remember that if you had an aggregate of size J and you add a monomer to it, that aggregate is no longer going to be of size J, and so you have to remove it from the bookkeeping. And so if you go through all these different steps, and you account for how they affect the concentration of aggregates of size J, you end up with this set of equations which look a bit complicated. And so the way to deal with this complicatedness is to say, well, actually, typically in experiments, what people are interested in are the average concentration of these aggregates. And so these quantities are typically related to the principal moments of the distribution. And so the lowest moments are effectively the aggregate number concentration, Ca, and the aggregate mass concentration, capital M. And so these quantities, because of the summing of the master equation with size J, satisfy a lower dimensional set of equations, which we call moment equations, and are shown here below. So if you sum the master equation over J, you end up with this new set of equations, which have a very straightforward physical interpretation. So the first equation for dm dt tells you that you consume monomers because of the growth of existing aggregates. And the second equation tells you that you form new aggregates, so you change the concentration Ca, either because of primary nucleation or secondary nucleation. And here I should maybe make or say as a technical point that in the secondary nucleation term, fragmentation is included as a special case when the reaction order N2 is set to 0. So in this case, you see that the corresponding rate term has a dependence only on the aggregate concentration. So now, as the second step, we need a liquid-liquid phase separation. And so here I'm going to consider a system of volume V, which contains a condensate. This condensate is formed from liquid-liquid phase separation between two components, A and B. A is the droplet material, so it's some protein material, and B is the cytoplasmic material, and it could be another protein, lipids, or water. Now, the key quantity here is the volume fraction. So how much volume of the system is occupied by species A or B. And the volume fraction of A, I'm denoted with phi, and then 1 minus phi is the volume fraction of B. So volume fraction 1 means I have only A, and 0 means I only have B. Now, in such a system, what drives phase separation? Well, according to the standard theory of phase separation, it is driven by two effects, opposing effects. One is the tendency of components to mix, and the other one is the interaction between these two components. And so this is what is captured in the famous Flory-Huggins free energy. And here I'm just, for sake of simplicity, focusing on a symmetric solution where A and B occupy the same molecular volumes. And so the mixing entropy term tells me how many ways I have in which I can arrange my components A and B. And the interaction energy tells me what is the relative interaction between A with A, B with B, respectively, A with B. And so the idea is that A and B always want to mix in order to increase entropy. But if the interaction between A and B is sufficiently disfavoring, this tendency is going to be counteracted, and so the system is going to phase separate. And so the key parameter that captures this relative interaction between the components is the famous chi parameter, which is the Flory-Huggins parameter and is set by the difference between the interaction energies of the different species. And so if this interaction parameter is small, you will see that the free energy is convex, and so the mixed solution is stable. But if you increase this parameter above a certain threshold, then you have a part of the free energy that is no longer convex, but is concave, and that is a sign of instability, and so that means that the system is going to phase separate. And so typically, you capture this behavior in terms of a phase diagram, where you look as a function of the chi parameter, how the free energy looks like and what are the regions of the free energy that are unstable. And so as you increase the chi parameter, you leave behind this kind of greenish region where you have phase separation. And if phase separation happens, the system will phase separate into two compartments, say an inside and an outside. The phase one is going to be composed mostly of component A and a little bit of B, whereas the surrounding component 2 is going to be composed mostly of component B. Now, to this picture, we haven't considered the role of the monomers. So now let's combine phase separation with aggregation. And so we have to ask, first of all, what happens if we add monomers into this picture? And the monomers now are going to play the role of the clients. And so the way to do that is to extend the Flory-Hagens free energy from before to include for monomers. So we will have a monomer mixing term, and we will have also monomer interactions with the droplet material A and with the surrounding material B. Now, this leads to a three-phase or kind of three-component Flory-Hagens free energy, which is typically difficult to deal with, but we work typically in the regime where monomers are dilute. So typical concentrations of aggregating monomers give rise to volume fractions which are very small. And so that allows us to expand the free energy in terms of small volume fraction of the monomers. And by equating the chemical potentials to find the equilibrium phase separation, what you find is that the monomers are not going to impact the phase separation of A and B, that phase separate as if the monomers weren't there, but the monomers are slave to the phase separation of A and B, and they will partition differently inside and outside, depending on their relative interaction with the inside and the outside. And so the partitioning degree, so the concentration of monomers inside versus outside gamma, is given by an exponential of the relative interaction between A and B and the degree of phase separation. So now, that means that if we have to kind of combine liquid-liquid phase separation with aggregation, we have to take two things into account. So the fact that monomers aggregate inside and outside according to the different mechanisms, and also the partitioning of the monomers set by the liquid-liquid phase separation. And so here we are going to work in a regime where we have a separation of timescales between diffusion of the monomers, which is assumed to be very fast, and aggregation, which is supposed to be very slow. And I'm going to assume that the aggregates do not diffuse due to their large size or the fact that they can get entangled with other polymers. So that phase, the separation of timescales means effectively that diffusion is so fast that if the concentrations of monomers inside or outside are going to change because of slow aggregation, the system will immediately react to reestablish the partitioning equilibrium, which is fixed by the laws of phase separation. And so that means that any deviation of the monomers concentration from the partitioning are going to be balanced by a diffusive flux of monomers, which I'm calling here J. And this flux comes from linear response theory, and you can show that in the linear response regime, this intercompartment flux is set effectively by the difference between the concentrations inside and outside compared to the partitioning degree. So it is zero, this flux, when the monomer concentrations are at the partitioning equilibrium. And so when setting up the equations for aggregation, we have to define concentrations of aggregates and monomers inside and outside. Monomers can aggregate because of primary nucleation, secondary nucleation, and growth. And I have this intercompartment flux that couples the inside and the outside. And this factor gamma, J, is exactly the key factor that is going to play a bigger role. So I'm not going to show you the solution, exact solution to these equations, but I wanted to show you a graphical solution for the aggregation. So imagine we start out with having a little bit more monomers inside than outside. So then aggregation is going to start initially inside because of the higher concentration. And so monomers are going to aggregate. The first aggregates are formed, they grow. And now the monomer concentration inside the compartment has dropped. And so there is now an imbalance in partitioning from the equilibrium. And so monomers are going to flow from the outside into the inside to reestablish equilibrium. And that will speed up the aggregation reaction even further inside and throw more material from the outside until eventually you are going to form most aggregates inside the condensate. And so to quantify this aspect, we have calculated the aggregate partition. So for example, how many aggregates you form inside the compartment versus outside at infinite time. And what we find is that this aggregate partitioning can be very large, as you would expect, due to this positive feedback mechanism between the outside and the inside, and is a power law of the monomer partition. So that's the input, and this is the gamma. The exponent of this power law depends on the size of the condensate. For small condensates, it is set by the reaction order of secondary nucleation N2 plus 1, and the plus 1 comes from elongation. Whereas for large condensates, the exponent is set by the reaction order of primary nucleation. And what is important here is to notice is that these reaction orders, for example, for the amyloid beta peptide, the factor N2 is equal to 2, so the exponent would be 3. And so even if you have a partitioning degree of monomers of, say, 10, that would result in 10 to the 3 partitioning of the aggregate. So it is really a strong effect, which is amplified due to this power law, which comes, as I said, from this coupling between the inside and outside from the monomer flux. And it's interesting here to note that this switch of exponent in the power law means that depending on the value of N2 and N1, having a small or a large condensate will yield a stronger enrichment of the aggregates. So for systems where N2 plus 1 is larger than N1, such as the amyloid beta peptide, for example, having a small compartments would lead to the maximal partitioning of the aggregates in the condensate. Whereas for other systems where N2 is small, for example, fragmenting systems like tau or dysprion, the prediction would be that a larger condensate would be more effective at localizing the aggregates. Just to kind of mention here at the end, not only you can control the position of the aggregates by playing with the partitioning of the aggregates, but you can also change other parameters of the aggregation, such as, for example, the total concentration of aggregates. And so what we find is there are regions of phase space, for example, in terms of volume fraction, where having condensates can yield either more aggregates compared to not having condensates at all, or yields less aggregates compared to not having the condensates. And more or less aggregates also couples with the average length. So that means we can also control the length of these polymers. And in fact, we can even control the entire length distribution. And this is recent work that I've been doing with a postdoc in Cambridge and with colleagues at the Max Planck Institute in Dresden. So with this, I hope I gave you a little bit of a flavor of what you can do in terms of using physics as a way to understand how these condensates can be used as controllers of biochemical reactions. Here I've shown you just example of a linear polymerization process where with very simple ingredients we are able to control, at least in theory, the position of these aggregates, their partitioning, their average size, length distribution, and so on. And in the future, of course, it would be very interesting to apply these ideas to a broader class of biochemical reaction problems. And with this, I would like to finish and thank all my collaborators, and in particular, Mahadeva and Thomas Knowles, who were my supervisors, and Christoph Weber for a great collaboration. And thank you all for your attention. And I'm very happy to take any questions.
Great, thanks a lot, Thomas. It was an excellent introduction. So do we have any questions? I invite people to unmute themselves or raise hands. So David, please go ahead.
Thank you, Thomas, for a very clear talk. I still like the story a lot, but I'm always a bit confused that you can model the aggregation process by simply basically writing reaction rate constants. And the energetics inside the droplet might be different than outside. And I don't know if that matters, or is it something you think about?
This is actually a very, this is a very good question. I mean, if I'm kind of to rephrase the question, I hope I understood it correctly. So you're saying, could it be that the rates are different inside and outside? This is, of course, the case. Yeah. So, of course, we wanted to, first of all, have a kind of proof of principle. So kind of we focused on the role of the concentration aspect alone, which I think was very interesting and very informative. Of course, in reality, the reaction rates would be very different. And also the diffusion properties are going to be very different. So what I can say is that from a small calculation, what you can show is that the effect on the rate constants has to be very strong in order to counteract the power law dependence. And so for at least exponents that we can get from experimental data, but of course they may be very different in reality. Really, you have to counteract order 3 power law or something like that, which is actually quite complicated unless you change the reaction rate constants by quite a bit, which could be the case, but definitely weak changes of the chemistry won't have such a huge effect as the power law coming from the coupling of the to the monomer flux.
Oh, thanks.
Well, thank you, Thomas.
Okay, fantastic, spectacular talk, Thomas. Really beautiful stuff. And this eLife story, I think, is just so exciting. One of the things I was sort of wondering is for many of the functional systems, for example, if you think about sort of tubulin nucleation and centrosomes and things like that, and I think there's similar phenomenology for actin. I guess you find sort of a hybrid scenario where sort of the role of the condensate seems to be to favor nucleation, but actually the growth of the fibers then happens in the dilute phase. Is that sort of a regime that emerges from this theory? And can you tell us a bit about when we might expect to see sort of that kind of regime?
Yeah, so this is actually a very good question. So we haven't really explored this regime directly, particularly because we have assumed that the main driver of the aggregation process comes from these secondary processes, so either fermentation or secondary nucleation. And so setting those terms to 0 might affect what the outcome is. So I don't know that question. And also there is, I think, a question about activity too, right? So most of these processes are also require energy, which is also an aspect that is not accounted in the theory. Yeah, so this is actually a good point, particularly because, I mean, if you think about, this enhancement effect coming from the coupling between the inside and the outside, the main driver of that is elongation, in fact, because this is the main consumer of monomers. And so I think it's a really interesting question to kind of investigate how that would change in a system where these secondary processes are not happening. Yeah.
Exactly. It's totally fascinating, right? Because you can imagine a scenario where with a really high primary nucleus size and no secondary nucleation, you might get a massive enhancement of the nucleation rate, but then the growth rate might be roughly the same inside the compartment and outside.
Exactly. Yeah.
Fascinating.
Yeah.
Thank you.
Thank you.
Well, thank you. Kafa, please.
Thank you, Thomas, for this very nice talk. So I have a question. So how do you think your theory can be modified, or as it is, explain how, so you see experimentally, let's say some protein can exhibit like phase separating properties, and then they can also like aggregate and form like fibrils that are forming from like scaffolding proteins, and like they start like nucleating on the surface and then they grow. So do you think your like this like theory can explain such systems, or like how can you modify it to explain such systems?
Yeah, this is actually a very good question, because I mean, of course, you are absolutely right. So effectively, the key assumption in this model is that effectively what aggregates are the clients. So it's not really the scaffold of the compartment, but in fact, in many cases, the scaffold itself can aggregate, and this is the case for many proteins that phase separate and then form amyloids as a result, of course. And so this theory as it is doesn't apply to these systems. But I think it would be very interesting to understand what aspects of these kind of changes when the reactant itself is the scaffold, or at least one of the reactants is the scaffold.
Thanks.